This diagram is the typical tropic HB axis, and it shows that the hypothalamus releases the releasing hormone to the anterior pituitary gland, which in response releases its tropic hormones to certain target glands. The product of these target glands are then released and serves as the negative feedback going towards the anterior pituitary and the hypothalamus. In the case of the HB gonadal axis, the releasing hormone from the hypothalamus is the gonadotropin releasing hormone, or GnRH, for both male and female. For the tropic hormones, for the male, it's actually the same. It's both FSH and LH, though they vary in the nomenclature for the LH. Okay? So for both male and female, the follicle stimulating hormone is known as both the FSH, but for LH, in males, it is called the interstitial cell stimulating hormone, or ICSH. And for the female, it's simply known as luteinizing hormone. Although in some cases, luteinizing hormone is also used to refer to the male tropic hormones. Then, of course, it would vary in the target cells for the target organs. For the testes, FSH affects the seminiferous tubules, and the LH or ICSH affects the interstitial cells of Leydig. In the meantime, for the female, FSH affects the ovarian follicle and it stimulates ovarian development, and LH influences the ovarian follicle, which in turn causes ovulation. Okay. So GnRH is the key regulator of the HB gonadal or reproductive axis. The cell bodies that releases GnRH are located in the medial preoptic area, or POA, and in the arcuate or infundibular nucleus of the hypothalamus. There are two modes of secretion of GnRH. There's the pulsatile or episodic release, and surge, which occurs in females during ovulation. But in males, there's no such thing as a GnRH surge. Release of GnRH is regulated by the recently documented or recently described kisspeptin unokinin B dynorphin, or KNDY neuronal network, or this kisspeptin signaling pathway. This is the most potent GnRH secretagogue. In males, it potentially stimulates the release of LH, but in females, its effect depends on the phase of the reproductive cycle. So you have to remember this kisspeptin. This is a relatively new hormone, more or less 5 years, or less than 10 years. So this is the most potent regulator of GnRH. Then for FSH, it is synthesized and secreted by the gonadotropes of the pituitary, anterior pituitary in specific. Stimulates the maturation of ovarian follicles, and it is also critical for sperm production. It stimulates primary spermatocytes to undergo meiosis. Then it stimulates the Sertoli cells to produce androgen-binding proteins, which is the main transport protein of testosterone or other androgens. And it is synthesized and secreted by the gonadotropes, the luteinizing hormone. Then ovulation of mature follicles is induced by the pre-ovulatory LH surge. So this also similar to GnRH. It also exhibits surge. And the theca cells and the ovary respond to LH stimulation by secretion of testosterone, which is converted then into estrogen by granulosa cells, which contains the enzyme aromatase. So this is required for the continued development and function of the corpus luteum. And in males, it stimulates synthesis and secretion of testosterone in Leydig cells. So that's the effect of the LH, or in males, it is called the interstitial cell stimulating hormone. Now we go to the male sex hormone, which is actually just testosterone. It is primarily synthesized in the Leydig cells of the testes under the control of the LH, which is called ICSH, as we have been mentioning. It acts on the Sertoli cells to stimulate spermatogenesis, and it is responsible for much of the female secondary characteristics, like for instance, changing in voice, growth of hair in the skin. So it is carried in the blood by the sex hormone binding globulin, and it interacts with its receptors as itself or in its reduced form, which is 5-alpha dihydrotestosterone or DHT. Okay? So it can act as itself, and it can also interact with its receptor as 5A DHT. Its anabolic effects include increased muscle mass and strength, bone mass, density, and strength. Then androgenic effects include primary development of male genitalia, increase in libido, secondary characteristics such as voice change, growth of hair, and plumage. This is you will highly appreciate the changes in the plumage and in avian. Now we go to the female sex hormones. Estrogen synthesis starts via the testosterone of the theca cells through LH stimulation, as already mentioned. Testosterone is converted into estrogen in the granulosa cells by the enzyme aromatase. The three main types of estrogens are E2, E3, and E1. E2 is estradiol, which is the most common type. Estriol is the main type during pregnancy, and estrone is the only type after menopause. The functions are mainly carried out by nuclear estrogen receptors, such as estrogen receptor alpha and beta, which functions as ligand-dependent transcriptional regulators. Both receptors are expressed in the brain. So this shows that estrogen has great influence in the behavior of animals. They also bind to estrogen response elements, or ERE, in DNA and recruit protein complexes that either enhance or repress transcription. So, as you may recall, in our early discussions in general physiology, we mentioned already that sex hormones are fat-soluble hormones, and as such, fat-soluble hormones mostly have the mechanism of action of regulating transcriptions because their receptors are actually intracellular. And in this case, estrogen has nuclear receptors. The most important function of estrogen is to prepare the endometrium for implantation. It promotes sexual receptivity during estrus, and it also induces LH surge, which in turn stimulates ovulation. Okay? So, in conjunction with the growth hormone, is secretory factor 1 or IGF-1. It is critical in mediating breast development during puberty. So, in preparation for gestation and subsequent parturition and lactation. Now we go to progesterone. Progesterone is secreted by the ovarian follicular cells prior to ovulation. It is secreted in large amounts in the corpus luteum that develops after ovulation, and it will grow from 10 to 12 days and then regress if fertilization does not occur. Okay, so progesterone is secreted by Cl, which is considered to be a temporary endocrine organ because it only forms after ovulation. Then if no fertilization occurs, it will regress. In the usual female reproductive cycle, progesterone prepares the uterus for pregnancy by shifting the endometrium from proliferation to secretion. Withdrawal of progesterone in the absence of pregnancy leads to organized shedding. In the case of primates and humans, menstruation occurs. It helps mediate sexual response also in the brain. After fertilization, it organizes the vasculature of the endometrium in preparation for implantation. It promotes enzymatic digestion of the zona pellucida to allow oocyte to implant into the uterine wall. After fertilization, it also inhibits contractions of the uterine myometrium and counteracts the effects of oxytocin on contractility. So in the presence of progesterone, oxytocin cannot affect the endometrium. It cannot cause contraction. It also promotes lobulo-alveolar growth in the breast in preparation for lactation, but it suppresses premature milk protein synthesis prior to parturition. So that's it for the HP gonadal axis with mentions of the male and female hormones. Our next lesson will be male reproduction.